Welcome back. This is going to be our second screencast for section 11.2, and if you remember from section 11.2 part 1 we had looked at one trait crosses, and so our focus for this screencast is going to be two trait crosses and the idea of independent assortment. So back in the 1800s, after Mendel's work with the garden peas, he had come to the conclusion that alleles do segregate. Now remember, the word segregate actually refers to separation. Now remember, we have two copies of every gene that gives us the characteristics that we have. One of those copies came from our mom, and one of those copies came from our dad. Now he understood, again based on his experiments, that those copies are going to separate and move into the gametes. And remember, the gametes are the sperm or the egg that's being produced by that individual. So if you remember our work with the punnett square, now this is a 16-box punnett square, but we had worked with the 4-box up to this point, that the items that you would put at the top and the left-hand side of that punnett square, these represent the alleles that you would find in the gametes. In other words, these are the possible combinations of alleles that might make their way into the offspring if fertilization did occur. Now, his next question was, he was curious, did segregation of one allele actually affect or influence the segregation of another pair of alleles? So what he's looking at basically is this: if you look at two characteristics in an individual, do they affect each other as those alleles travel into the gametes, as they travel into the sperm or the egg? So what he did is he decided to follow two different genes, basically genes for different traits from one generation to the next, and that's what they're doing over here on the right hand side. So if you notice, we're looking at two characteristics for these peas. We're looking at color, so we have a yellow versus a green, and we also look at the texture of the pea. So it's either smooth, sometimes called round, or sort of a wrinkled type of pea. And we've assigned a different letter to each of those characteristics. If you notice, the R's are going to basically represent the texture of the pea, and the Y's are going to represent the color. And just like we had done with the M1 trait crosses, we set up our punnett square, but this time we actually have 16 boxes because we're working with two traits instead of just 4. And we set up our gametes, again those sex cells, those sperm or egg, on the top and the left-hand side. Now, we call these a two-factor cross, and the 2 is referring to the two different traits that we're looking at. So the first thing that he decided to do is he decided to cross two true breeding individuals. So this is going to represent our P generation. So this is our first initial cross. And they're true breeding, and if you look here, this is a true breeding individual because they are homozygous for that particular trait. And if you notice, this is homozygous recessive, so this is going to give us a wrinkled type of texture, and it's going to be homozygous recessive for the color as well, so it's going to give us a green pea. Now down here, again, we have a true breeding individual because they're homozygous for both genes, but in this case they're homozygous dominant for both, so they have capital letters for both alleles. And again, if you notice, when we're looking at the texture, the dominant allele is going to be the smooth or the round pea, and in this case the color yellow is going to be the dominant color in this combination. Now when he made the cross, in other words, he put his gametes on the top and the left hand side, the possible gametes that we have here, he found that the offspring that were produced were all yellow and they were round or smooth peas. All right, so this is going to represent our P generation. In other words, these are our initial cross, our initial parents, and the F1 generation is going to be the offspring that you see being produced from this combination. So the next thing that Mendel wanted to do is he wanted to take two of those F1 offspring from the previous screen and actually cross them and see what the outcome would be. So if you notice, we have our very first individual right up here, and again this was taken from that original parental cross, so the offspring that were produced from that P cross that we had done before. And if you notice, they are heterozygous for both traits, so heterozygous for the texture of the pea and heterozygous for the color of the pea. And again, if you notice down here, this one is yellow, this one is going to be smooth, again heterozygous for both of those characteristics. So just like we have done before, he went ahead and set up his gametes, and so if you notice, these are the variety of gametes that are being produced from this combination, and these are the variety of gametes that could be produced from this combination. And then just like you would do with a one-trait cross, you simply bring these two sets together, and that gives you the genotype of that particular possible offspring. So if you notice, the expected ratio that we should get phenotypically, remember phenotype refers to what you would see, is going to be a 9 to 3 to 3 to 1 ratio. Now the way that he actually got that, if you look at the possible offspring on the right hand side, you're going to notice that we have 1, 2, 3, 4, 5, 6, 7, 8, and of course 9. We have 9 that are smooth peas or round peas, and they're yellow in color. Now if you notice, we have 1, 2, 3 that are going to be smooth or round, but they're going to be green in color. Then we have 1, 2, 3 that are going to be wrinkled and yellow in color, and then of course we have one down here, this very last one in the corner, that is going to be green and wrinkled. So that's where we get our 9 to 3 to 3 to 1 phenotypic ratio. So we're not talking about the genotype arrangement here, we're talking about what you actually will see. What would be the phenotypic result? So based on this ratio and the data that he looked at, he noticed that this data actually suggested that independent assortment actually did occur. So what that means is that each of these different alleles, each of these different traits or genes actually segregated independently during the formation of the gametes or the sex cells, basically meaning that there was no influence of one over the other. Now that was a pretty quick overview of some of Mendel's work with his two-factor crosses and the idea of independent assortment, but what we need to do is we need to make sure that you understand how you need to set up a Punnett square if you are given a two-factor cross, which you will, to do in class. So what we're going to do is we're going to do a couple of practice problems to make sure you understand how to get this started. So if you notice in this particular first cross that we're going to do, in pigs, curly tails, capital C, are dominant over straight tails, which is going to be your lowercase c. Now the pink color, capital P, is going to be dominant over the gray color in these pigs, which is going to be your lowercase p. So in this case, what you're supposed to do is you are supposed to cross a male pig that is homozygous dominant, so again you got to read your problems very carefully for both traits, and a female pig that is homozygous recessive for both traits, so both the type of the tail and for body color. Now, as I had said in class, the very first thing you want to do is you want to set up your parental cross. In other words, what are the genotypes of each parent? So again, the male pig was going to be homozygous dominant for actually both traits. So we have 2 big C's and we have 2 large P's. So that's going to be the genotype for that male parent. And we're going to cross that with a female that's going to be homozygous recessive for both the type of the tail and again the body color. So what we have is we have 2 very small C's and 2 lowercase p's. Now this is a situation where we're using letters that it might be a little bit difficult to tell which is the capital and which is the lowercase. So again, make sure that you exaggerate the size of the letter so I understand what you're working with. So this is going to be our very first step. This is going to be setting up our parental cross. So then what we're going to do is we're going to go ahead and bring this over to the right, and I'm just going to go ahead and put the genotype up here on the top, and I'm going to put the genotype for the second parent over here on the left. All right. Now this is where things sometimes become a little bit difficult. Sometimes people have problems knowing exactly how to put the gametes along the top and left hand side of their Punnett square. So I'm going to show you. So what you need to do first is you need to take this first C and you need to connect it up with that very first P. And so that gives you a large C and a large P. Then you're going to take that first C again and you're going to connect it to that second P. So again, large C, large P. Then you're going to take the second C, connect it up with the first P, again large C, large P, and then take that second C again and connect it up with that second P, again large C, large P. So that's going to give us one allele for each of the different traits that we're looking at. Remember, you can only pass one of those on to your offspring. So those would be the alleles that might be found in the gametes produced by that parent. Now down here towards the bottom, you do exactly the same thing. So we take the first C, connect it up with that first little P. So again, it's going to be our first set of possible gametes. And then you take that first C, match it up with the second P. Same thing. Then you go ahead and match up that second C with that first P. Again, same thing. And then the fourth thing is to match that first or second C, with that second little P. All right. So now again, you have the possible gametes that would be produced by that parent. Now, what's interesting about this Punnett square is all of the possible gametes are the same for each parent. In other words, there's no difference between those gametes. So what we're going to do next is we're going to go ahead and fill in our Punnett square. So we do the same thing we had done with a one trait cross. We take this set of alleles and we match them up with this set of alleles. So this is gamete 1 and gamete 2. So we bring them together, so we would have a capital C, lowercase c, capital P, and a lowercase p. Alright, so there are going to be a possibility of it being heterozygous for both of those different genes. So same thing we did before, take this set right here, match it up with this set, capital C, lowercase c, large p, lowercase p. So same thing over here, take this set, match it up with this set, capital C, lowercase, capital P, lowercase p. And then last, take this one and match it up with this one. Again, capital C, lowercase c, capital P, lowercase p. You would do the exact same thing for the next three rows. So this one would match up with this one would match up with this one, just work your way down. Now what you can do is, you'll notice that we had the same exact arrangement for all four of these, and that's because all the gametes up here are the same, and it's the same thing on the left hand side. So to do a shortcut, or to make this a little bit more efficient, if you notice that that's going to happen, you don't have to spend the time actually filling out the entire Punnett square. You can actually draw lines, and that makes it a lot simpler because you know they're all going to be exactly the same. Now I'm still going to, now that was step 2, I'm still going to ask you possibly to find the genotypic and phenotypic ratios. So that would be step 3. So if you notice, genotype is going to be the possible genotypic arrangements of the offspring, and if you notice we only have one possibility, capital C lowercase, capital P lowercase p. And so the genotypic ratio in this case is going to be 100%, which means all of them are going to be capital C lowercase, capital P lowercase p. There's no other possible combinations on the right hand side. Now the phenotypic ratio, again if you notice, this particular situation is going to give us again a 100% chance, in other words there's no other combinations here, of getting a, let's see, capital C, so we had pink color dominant over gray, so we're going to have a pink pig that is going to have a curly tail. So we have a pink pig with a curly tail. Again, it's 100% because you don't have any other combinations on the right hand side. So again, step 1, set up your parents. Step 2, fill out your Punnett square. And step 3, come up with your ratios if you are asked to do that. Alright, so this is going to be our second practice problem. And in this one it says in humans brown hair, capital B, is dominant over blonde hair, lowercase b. Unattached earlobes, capital E, are dominant over attached earlobes, which is lowercase e. Now what we're going to do is we're going to cross a male that is heterozygous for both traits. So we'll start off with that first parent. So capital B, lowercase b, capital E, lowercase e. Now we did that because they're heterozygous. Remember, hetero means different. So we have a different allele in that setup. So capital B and lowercase b. It's the same gene, but different alleles. And again, it said for both traits, so we know that for the earlobes, same thing. It's going to be heterozygous for that situation as well. So that's going to be crossed with a female that is heterozygous for both traits. So same exact setup. Capital B, lowercase b, capital E, lowercase e. All right, so that's going to be our very first step. Now the second step over here on the right is going to be to set up our Punnett square. I'm going to do the same exact thing I did before. I'm going to put my first parent right here, and I'm going to put my second parent down here. Now remember, these parents are going to produce a variety of gametes. Now remember, I had told you that in order to find out what the possible gametes along the top would be, we need to take that very first B, match it up with that very first E. So it's going to be big B, big E. Then we take that first B and match it up with that second lowercase e. And so we have a big B, lowercase e. Then what we do is we take that second lowercase b and match it up with the uppercase e. So we have a little b, and a uppercase e. And then we take that lowercase b and match it up with our lowercase e. So we have a small b and a small e. Now we're going to do the same thing down here with this parent. So what we have is we have an uppercase b, uppercase e. We have the uppercase b and the lowercase e. Then we have the second B, which is lowercase. It's going to match up with that uppercase E. Then we have our second B. It's going to match up with our lowercase e. Now this is a situation where the letters that we're using are very different from each other, so it's really easy to recognize the difference between our dominant and our recessive alleles. So now that we have our gametes set up on the top and the left-hand side, we do exactly the same thing we had done before. We're just going to match them up. So in this case, we have a big B, put those two together, and we have two big E's. Over here, you notice we have a big B, capital E, but we have a lowercase e right here. So again, we're bringing this one and this one together. Then we have a capital B, we have a lowercase b here, capital E, and a capital E. And then this last one, capital B, lowercase b, and then we have capital E, lowercase e. All right. And so then what we do is we'd go ahead and fill out the entire Punnett square, and then of course what we would do is we'd go ahead and find our genotypic and phenotypic ratios. Alright, so what I've done is I went ahead and completed our Punnett square, and so the third step, if you are asked to do this, is going to be to come up with your genotypic and your phenotypic ratios. Alright, now I'm not going to spend the time actually going through this, but what you need to know is that you need to do simply this. When you do genotypic ratios, you need to look at the possible combinations of the genotypes that you have in your Punnett square. So if you notice, we have one combination of big B, big E, big E. All right, and so what you do is you look throughout your entire Punnett square, and all of you can devise a way that works best for you to actually make sure you keep track of these, and you see, do you have any arrangement that is the same as you see right here? Well, if you notice, there's no other possible arrangement that looks like this. In other words, we only have one big B, big E, big E. And so I would say 1, big B, big E, big E. And so the next thing we would do is we would look, okay, this next one, big B, big E, little e, how many of those do we actually have? And if you notice, we have 1, and there's 1 right here, so we have 2. So again, we kind of scan through our Punnett square and to see if we have any other situation where we have 2 big B's and a big E and a little e, and see if that occurs anywhere else. And if it doesn't, then we know that we have 2 big B, big E, little e. All right? So this is a situation where sometimes it's easier just to work with the number of boxes that you have and not really the percentages or the fractions that sometimes I said you could use if you wanted to. Now, phenotypically, same exact thing. We're looking at the expression of that genotype. So what you would do is you would look at this first one, and if you notice, this one's going to have brown hair and unattached earlobes. That's what's represented by these dominant alleles. Now, if you notice, that's going to be, we'll go ahead and maybe use a little circle or a dot right there. That gives us that particular combination. This one gives us brown hair and unattached earlobes. This one gives us, again, brown hair and unattached earlobes. This one gives us brown hair, unattached earlobes. This one does the same thing. This one does the same thing. This one down here on the lower left gives us, again, brown hair and unattached earlobes. And then if you notice, this one does too, and this one does too. And so then we do a quick scan throughout our Punnett square if there's any other situation where that would give us that phenotype. And if you notice, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. We had 9 of those. So we have 9 that will have brown hair and unattached earlobes. All right? So then what we do is we go ahead and look at the next one. So if we look at this one's going to have brown hair and attached earlobes. And so we maybe put a little X right there, so that gives us brown hair attached. This one right here gives us brown hair attached, and down here, brown hair attached. And what we do is we go ahead and look and scan. Do we have any other situation that would give us that phenotype? And if not, we go ahead and count it up. 1, 2, 3. And so we have 3 that would be brown hair, and this time it would be attached. Now, we go ahead and look at the next combination. So in this case, we look at maybe this one right here. So this one has blonde hair, because remember, blonde was recessive, the lowercase b's. blonde hair, and actually unattached. And so we'll put a little star here to represent that one. And this is going to be brown hair, or excuse me, blonde hair and unattached. We'll put a star here. And this one's going to be blonde hair and unattached. All right. So then we go ahead and say we have 3 of those. So we do 3 blonde and unattached. And if you notice, we have 1 left right down here. And so we'll go ahead and just circle this one. This is the only combination that we have left, and this one is blonde hair and attached earlobe. So we have 1 that is blonde and attached. All right. Now, if you recognize, this is the same ratio that Mendel had come up with his garden peas. We have a 9:3 to 3:1 ratio. All right, because again, both parents were heterozygous for each of the traits. So that's how you would come up with your phenotypic ratio. Now, most of the time, I'm going to be asking you probably just for the phenotypic ratio on an assessment, but do be prepared just in case. I might also ask you for the genotypic as well. So remember, phenotypic is what you see. Genotypic is going to be the genetic arrangement or the possibility for that particular offspring. Alright, so that's going to finish up our second screencast for section 11.2. As always, please make sure that you have completed your screencast notes before you come to class.